we're going to talk about the evaluation of AAV transduction of CHO cells. And then Isso is going to talk about HEC293 cells and some of the analytical assays associated with that production. So we're going to start with Dominique Ingato from Mira Costa College. Hi, Dominique.
Hello, everyone. Thanks for being here today. We can go ahead to the next slide, and I'll talk a little bit about the experiment that I developed as part of this project. So for this experiment, we were transducing CHO cells to be able to express green fluorescent protein using AAV. And in order to assess this, we use flow cytometry, which is a really crucial skill that students should be at least learning about to some extent, and in the best case, have hands-on experience with. In talking with our industry partners, we found that a lot of them were really excited about the idea of students coming in with flow cytometry knowledge, because it is a bit challenging to find candidates who have that experience, and it is something that's used frequently in gene and cell therapy. These companies included Kite Pharma and ViaCyte, as well as others who are really excited about having students be able to do flow cytometry. So as we continue on, I'm going to show a little bit about the instrumentation, Maggie, on the next slide. So for this experiment, we needed a flow cytometer. We used this Luminex Guava Easy Site, which we were really lucky to be able to set up, I think it was last school year, as well as a cell counting Countess 2. You could do this manually using a hemocytometer as well, but we used the Countess for cell counting. And then, of course, since we were using CHO cells, we needed a lab that was equipped for mammalian cell culture as well. So if you don't have access to this equipment, there are always modifications. There's a Labster with flow cytometry. There's ways that you can partner with other schools that may have this so that you can get data to show to the students. And I would be happy to work with you on that if you don't have a flow cytometer available. So let's go ahead and we'll continue on, and I'll tell you a little bit about the experiment. And we can click to the next slide, Maggie. And I apologize before that. Before I tell you a little bit more about the experiment, I'll tell you a little bit about the industry applications here. So we've already talked a lot about the background into how cell and gene therapy work. And for gene therapy in particular, I really like this roadmap that shows you how to get all the way from the early stage research up to the complete product. And so one example that I highlight here is the in vitro characterization of AAV, whether or not the cells have been transduced and whether they're actually expressing that protein of interest. And flow cytometry is one method that we can use to do this, and it's commonly now being used more and more in industry as gene therapy is growing. And Maggie, we can click to the next slide and take a look at cell therapy. We talked a little bit about CAR T cell therapy earlier, and this graphic does a great job at showing how from early stage, collection of that patient-specific material all the way through manufacturing and, characterizing that product, and then long-term patient monitoring, we can use flow cytometry to be able to assess whether the cells are, producing that protein of interest and expressing that. This is essentially used at all of those different stages. So again, it's an important technique that you would want your students to at least be familiar with, and in the ideal case, have some hands-on experience with as well. So Maggie, we can click to the next slide and we can go ahead and look at a brief summary of the procedure. I do have a full-length SOP that's available, and I will include my email at the end of my portion of the slides. But this is just a brief overview so you can get an idea of what this experiment involves. So the first thing to do for this experiment is to prepare those cells for transduction. So I used media, F12 ham media with FBS and gentamicin. And I used AAV for the transduction of eGFP. And I have a whole protocol on this, but essentially I needed to dilute this out prior to adding it to those CHO cells. The CHO cells were plated in 6-well plates so that I could test several different concentrations of my AAV. And we'll take a look at those results later on as we get to the results section. But you can see as we move on to the transduction and preparation for assessment that there were several different concentrations tested. These were then incubated with the cells for 48 hours. And then the cells were trypsinized, and we assessed viability as well as the transduction efficiency. So like I mentioned, for viability assessment, I used the Countess device. But again, you don't need to have that if there's another way that you assess viability in your labs. You could do this manually with a hemocytometer as well. And Maggie, if we click to the next slide now. So for our transduction efficiency, this is a little bit more complex. And again, this is just a brief summary, so there are many more steps. But using the flow cytometer, you have to start off by cleaning the instrument and performing an easy check. So essentially making sure that the instrument is functioning as expected. Oftentimes you can purchase easy check beads that will allow you to determine if the instrument is in fact counting all of the beads that pass through it, as you want it to count all of the cells that pass through and come up with an appropriate concentration. And if you find that your concentration falls outside of the boundaries of 10% of what is expected, you know that there may be some type of an issue there. From there, once that is all run, you can go ahead and run your transduction efficiency test by taking those trypsinized cells, resuspending them, and then running them on the flow cytometer. So the flow cytometer will take samples of those cells and it will individually pass those cells through the capillary, look at the fluorescence. In this case, we were looking for green fluorescence for our GFP. And I'll show you as we move on what this data actually looks like so we can look at how we actually analyze that. So we can go ahead, Maggie, to the next slide. So here's an example of some flow cytometry data. This was our control where we did not add the AAV at all. And on the left-hand side, we have our side scatter versus forward scatter graph. Just as a very brief overview, if you have not done flow cytometry before, this chart is telling us a little bit about the size of the particles as well as the granularity of the particles that are passing through. So for our cells, we expect them to have similar characteristics. I've actually circled where those cells are showing up. And any particles that might be a little bit bigger or smaller could be debris, could be cell debris, could be some other type of contamination. So we want to specifically select that cell population, and then we're only going to analyze the results from that cell population. So we're going to then, for our graph on the right, we're going to apply a gate so that we only look at those events or those cells that showed up in the circle. On the right, on the x-axis, we have our amount of green fluorescence, and then on the y-axis, we have the cell count. So you can see that we have a nice single peak here, indicating that our cells, all fell within this single peak. And I can then add some stat markers, which are the red lines. And those red lines will allow me to determine the percentage of events that are falling within them. So you can see here that the line that goes on the left portion of that graph contains about almost 100% of the events. So pretty much 100% of my events were characterized as not being fluorescent, whereas a very small percent, pretty much negligible, was characterized as having a higher amount of fluorescence, which we're going to say, is what we count as having been transduced. So if we click to the next slide, we can now compare this to a case where we actually did add the AAV. And this was the highest concentration that was tested. And we can see now that the same process was applied, but we now have fluorescence showing up on the graph on the right-hand side. So now 85% of our cells are being transduced based on how the stat markers were set. And we still have maybe about 15% that are not hitting that region of being, producing enough GFP to show up as being fluorescent. So this is, a typical experimental result that you would get from flow cytometry. And again, if you don't have a flow cytometer in your lab, I'm happy to provide you with graphs that you can talk students through so they can at least get that experience as well. So if we click to the next slide, we can take a look at the transduction efficiency using different volumes of AAV added to the wells. And we can see that the control, obviously it had a very low percentage of the cells showed any green fluorescence, whereas for all of the concentrations, we had relatively high transduction efficiencies, and something that would be interesting for the students to look at. So if you're planning to do this experiment and you have limited reagents, the AAV is somewhat expensive, you can use as low as 10 microliters and perhaps even lower than that. I did not test lower concentrations, but that's one way to save a little bit of funding there. And with that, we can go on and continue to look at a few more results. So one thing that I did want to make sure that we checked here was whether or not our cells were, surviving this process when they were transduced with the AAV. Were they still showing the similar levels of viability compared to the control? So this is a screenshot from our count test for the control where we didn't have any AAV added. And you can see that about 79% of our cells are viable. And then on the next slide, we'll go ahead and take a look at the viability across the different wells. And you can see that we have fairly consistent viability here. I didn't do multiple, I didn't do multiple versions of this experiment, so we don't have any error bars here, but you can see that we don't seem to have any major changes in viability, which is a good thing because we wanted to make sure that our cells weren't being killed during this transduction process for any reason. So with that, we'll click to the slide right after. And overall, we were able to demonstrate that these CHO cells could be transduced with AAV-GFP. And the flow cytometry was a way to measure that transduction efficiency, just like they do in industry. This is something that I'm really excited to bring into the lab at MiraCosta. We do have our flow cytometer up and running, and this will probably be introduced to our students, if not this semester, at least the following semester. Like I said, there is a very detailed SOP that is much more detailed than what I showed you today. So if you're interested in that, please shoot me an email. My email is listed on this slide. And with that, I can pass it on to the next presenter, or Maggie, if you want me to take any questions, I can do that as well.
Barbara, do we have questions?
I don't see any questions right now.
All right, so we'll continue on and maybe we'll get some. So thank you, Dominique, for that. And from what we learned in our SME listening sessions, flow cytometry is one of those skills and experience they want our graduates to have. But as we went through and we're developing this curriculum, we're thinking of other ways for programs to adopt some of the cell and gene therapy experiments. And if you don't have, like we don't yet have a flow cytometer, how can we introduce some of these concepts? Can we transduce CHO cells with AAV-GFP and analyze it by other methods? So whereas Dominique took the high-tech approach with our flow cytometer, Hetal and I at Montgomery County Community College looked at transduction simply using fluorescent microscopy and also Western blot. So if you don't have a flow cytometer, you can do this experiment with sort of a less sophisticated tools. So we followed, similar to what Dominique did, we grew CHO cells on a 6-well plate. We purchased AAV2 GFP, so recombinant adeno-associated virus serotype 2 from Vector Builder, the same product that Dominique used, and we incubated and transduced CHO cells with that. After 48 hours, we evaluated the level of GFP expression under a fluorescent microscope. We also prepared cell lysates using typical RIPA buffer preparation of a lysate. We ran an SDS page and we analyzed GFP expression with Western blot. So this is what our cells looked like after 48 hours. This is a 10x magnification. You can see many cells have been transduced and are expressing GFP, but there are still obviously some we don't have 100% transduction. And then at higher magnifications, this is 20x magnification and 40x. So we can see some non-transduced cells, and we can see some very bright GFP producing cells as well. So this was a quick look to see if the transgenes would actually enter the cell and got expressed, and the protein was made, and the protein is fluorescing. Again, we made lysates from these cells after 72 hours. These are our lysates here. So this is our control non-transduced lysate. It's slightly overloaded, as you can see. We've done this experiment just once. We still have to optimize it, but you can see we have 3 pretty evenly matched cell lysates with control and 2 transduced cell lysates. And here's pure GFP that our students produce using IMAC chromatography. So this is a his tag GFP. the EGF, his tag EGFP, the EGFP and the AAV viral vector is not his tag. And then we have our Western blot, and as many of you may know, GFP is about 26 kilodaltons, and here's our sort of a massively overloaded pure GFP, and we have a couple of non-specific binding bands from our antibody. But here we can clearly see GFP production in both of our transduced cell lysates, but not on our control cell lysate. So again, sort of a more sort of straightforward way using techniques that we already have incorporated into our program of introducing gene therapy transduction of cells. So we used an anti-GFP monoclonal antibody, and we used an alkaline phosphatase conjugated secondary antibody for this colorimetric reaction. So this is our low-tech approach to evaluating CHO cell transduction by AAV GFP. And I'll pause there.
There was a question in the chat. Clarification, are these transductions all non-germline cell transductions? And then I think Jim answered that. True, they are somatic cells.
Yeah.
Yeah,
So
I'll take it. I'll add to that the different serotypes, the different strains of adeno-associated virus will target different tissues. And the different tissues, whether it's the liver or the eye, they're all somatic cells, no germline.
So make sure you match up your AAV serotype with the cells that you want to transduce, as Jim is saying. So with that, then we'll move on to Isso Bayala from Quincy College, who's going to talk about HEC293 cell production of AAV2. Isso, it's all yours.
Thank you, Maggie. Hi, everybody. So this is the continuation of the first part. So I'm going to do a quick summary of what we did in the first part. Then we're going to move to the quantitation of the virus particles. Yeah, please, first slide. Yes, this one. Yeah, so here is, we purchased this from Thermo Fisher, the media and the cell line. So there is a media and the feed. The glutamax is a feed that we add to the production media with a concentration of 4% to make the culture medium, the complete medium, and with a volume of 30 ml. So we dump the entire cryovial, the content of cryovial into the 30 ml. So then I put it in an incubator, incubator shaker with CO2. Next slide, please. Yes, here. So incubator shaker with the parameters on top, CO2, 37 degrees, and the agitation is 125 RPM. So it stays there for until we do the cell count. Yes, you're right. Next slide. Yeah, we monitor the cells and count the cells. Sometimes they take about 3 days, sometimes 4 days. Sometimes you have to centrifuge, collect the pallet, and change the media. So all of these happened when I was doing this. And one time I got a contamination and they had to do it over. So Thermo Fisher developed the cell line, and they recommended that the transfection is good when you have a cell density that's between 4.5 and 6 million cells per ml with a viability of 95 or higher. So we were in that range. Then we did the transfection. Next slide, please. So 3 plasmids are used to transfect the cells, and we purchase those cells, those plasmids from through Thermo Fisher from a company called Cell Biolabs. So it's ready to go. We mix. There is a procedure. We mix the 3 plasmid, then we transfer everything into the shaker flask together with the cells. Next slide, please. Yes, so this is the cells, cell culture transfected. So then we wait for 72 hours, about 3 days. Then if it's successful, the cells will be transfected. So they're gonna incorporate the virus. The virus is gonna actually infect most of the cells. So then it's recommended on the next slide, please. So after the 72 hours, we can directly add the lysis buffer into the cell culture, lyse the cells like that, and wait after 1 hour. Then we continue with a harvest and centrifugation. That's on the next slide. So centrifugation, and in the supernatant, the virus particles are in the supernatant. So then the next step, that's where we stop for the next, the first part of this project. So the second part of this project, we did the quantitation of the virus particles. So up to here, we didn't know if we had virus particles or no. We just didn't know. We just followed the protocol. But there is a qPCR kit with the primers that are specific to AAV2. So we purchased that kit and we did a qPCR. That's on the next slide, please. So the qPCR instrument we use is called QuantaBio. It's a small and it's relatively easy to use. The software can be downloaded for what they don't. The software is available and you can just download it, and it's portable. So it's actually a good tool for teaching. So we use the QuantaBio. And on the next slide, please. So this is the protocol to prepare the primers and the protocol to prepare the samples. So the next slide, please. So we prepare the samples and the primers. Then we proceed with the method. The method is here. This is how the software looks like. And we use the protocol, and it's a two-step qPCR. So then we just plug in the numbers, and some people do 35 cycles. We did 40 cycles. And so you're gonna actually get your result before 40 cycles. You're gonna see that on the next slide. One more thing, we modify a little bit according to, with the help of the QuantaBio qPCR specialist. They recommended that we use cyber green, that instead of the dye that came with the kit. So we use cyber green. This kit. We modify a little bit the method to run the qPCR. So this is the cell lysate. After centrifugation, we take the supernatant and we run the qPCR. And if you look at the result here, you have two baselines. There is one red baseline at the bottom, and that's the standard, the positive control with real viruses, AAV2, known amount of AAV2. And if you look at the lines, the second baseline right around the middle, so those are the samples that of the supernatant. So we were told that it's not a normal. You should have one baseline for the samples and the control. But here the samples are a little bit of a baseline that is higher than the baseline of the control. And that was probably due to impurities, because the cyber green bind to other DNA molecule. If they are two-stranded DNA molecules, they bind, the cyber green bind and can give you a fluorescent detection. So that probably, that's what happened here. And next slide, please. So this is the integration of the result. After you run it, you can actually, the software gonna integrate the result and give you what is called a CT. The CT is the threshold of cycles. That's the red line there on the screen. The red line shows how many, the software needs a certain amount of material to be able to quantify it in a way that is statistically valid. So that's the red line, that's the CT. And there is also a CQ, which is, the CQ is the quantitation cycle. So if you look at the lines on that cross the red horizontal line, so you have one green and one blue. So what it means is for each line, it doesn't take, the blue line takes less cycles, that's around 12 cycles. And the green line takes about 17 cycles. So it means it takes, there was less material in the green before you get to the CT. So the CQ tells you how many cycles it took for each tube, each sample to reach the threshold, the red line. So this was with the unpurified sample. So that's what we get. So what we did next, we tried to purify the sample using Acta Pure. That's will be on the next slide, please. So we use the resin specific to AAV2, that's affinity resin, with a column, pack the column. And we use Acta Pure to purify the supernatant. And we got various particles. Here, if you're gonna do this, you wanna be careful because the protocol given by Thermo Fisher did not work well with me. I had to modify because I was collecting the wrong fraction. And when I ran the qPCR, I was not seeing any signal because I was losing I was not collecting the right fraction during the elution. So I did that a couple of times, and then I found out I collected all the fraction I could get, all the peaks, and I tested each of them. So I realized that the virus came in a fraction that I was not collecting. So that's how I found out. That will be on the next slide. After purification, after we were able to collect the pure fraction, and we run it again, and this time you can see the baseline. The baseline is the same for the standard and the samples. So it started the same, but you can see the first graph that is higher comes up is a standard, it's more concentrated, and the other ones are less concentrated, so they takes them more cycles to be detected. But this is not, that's what you see on the screen when you run it, but this is not an integration. The integration is the software gonna calculate how much material is enough before it can actually quantify it in a way that is statistically valid. So that will be on the next slide. Yes, so you can see here that below the red line is called signal baseline noise. So it doesn't, it's not, you cannot really accurately quantify your samples below the red line. So the red line is called the CT, the threshold, which is that line represents where valid to quantify. And the software does it by itself. And the software also gives the number of cycle for each tube, each color, each graph color correspond to a different tube. So the software tells how many cycles it takes to detect each sample. So then you could, from here, you can actually collect the CQs. The CQs, you can collect them and use that to plot a calibration as a standard calibration graph. Then you could use that to determine the amount of particles that were present at the beginning of the cycle. So what it means, each cycle is when the amount of product doubles. So the number of the particles, the quantity doubles with each cycle. So once the software is able to positively quantify the material, we could use that, and based on the number of cycles that were used to detect it, we can go back and find out how much material were there before we reach that number of cycles. So that's how the quantitation is made. Next slide, please. There is also an ELISA kit specific to AAV2. AAV2, so the qPCR is specific to all the AAVs, all the stereotype, but the ELISA is specific to AAV2. So we purchased that kit, and what we didn't have too much time to make many runs, but we ran this test three times, and we got almost the same result. So that will be on the next slide. So this is the plates. The plate comes with the kit, and the plate is coated. It has antibodies that are attached to the bottom. So we add the samples. So what we did, the kit also comes with a positive control with a real AAV that we can dilute, which we did in the first two rows. We did a serial dilution, and we duplicate each dilution, and we run it. And on the last row, what we did, the bottom row, we just put a different, the same amount of the purified sample. So to get many samples, many results that can be used to statistically calculate the average of the detection. This is a sandwich ELISA. The protocol is a simple, straightforward. You prepare the reagent, and there are a lot of wash and incubation. It takes about 2 hours. Then after that, we read the absorbance, the optical density at 450 nanometers. And based on that, we use the average of the reading that we got with the sample, the row down, and we use that, the first 2 rows, the serial dilution one, we use that to plot a calibration standard curve, which is on the next slide. So yeah, we have the calibration curve. Then since we know the optical density of our sample, we can use that with the equation to calculate the number of particles that are present in the sample that was purified. So that's how we did the ELISA test. I believe I think there is a last one. So the next step will be like Jim was saying and Maggie was saying, the capsids, some of them gonna be empty, some of them gonna be full. The empty one, for patient treatment, you want the full capsid. So it's better if it's possible to separate the full from the empty. And HPLC is one of the methods that is known to do that. We haven't been able to do it yet. We tried many times, but it has not worked yet, but I'm still working on it. So that's what we have done so far.
Thank you, Isso. Thank you, very nice. So we could just hold our questions if you have any for Isso till the end. So we have a chance to hear Barbara speak about our equipment recommendations from our subject matter experts. Barbara.
Thank you, Maggie. So first of all, everything you're going to see is on a document that you will receive. So please don't feel stressed to write anything down right now. Just let the information just wash over you. Next slide, Maggie. So I've just divided this, and this is exactly how the report is divided by the different unit operations. So upstream, downstream, fill, finish, and then process support. We're really talking about buffer media preparation, and then analytical testing. And I have the line to fill finish dotted because, as we heard earlier today, especially for cell-based therapies, that analytical testing really has to be sped up at the end. Next slide. So let's start with upstream. Again, a lot of this equipment is going to be very familiar to you. And if you have a biomanufacturing program, you probably have most of what you need. So you're just going to need some general cell culture equipment with incubators, biosafety cabinets, cell counters, even though you can use hemocytometers with microscopes. Go ahead and click again, Maggie. And then in terms of single-use equipment, this is where it might be a little bit different depending on your setup if you have biomanufacturing. Really in cell-based therapy, particularly, it's all single-use equipment. So we're looking at single-use bioreactors. They could be suspension or adherent depending on the application. If you click one more time, Maggie. And then we have aseptic connectors and tube welders, which Jim had alluded to earlier today. So really the main difference here is that the cell culture is going to be single use. Okay, let's move on. And then obviously control units for bioreactors. So in terms of upstream processing specifically for cell-based therapy, again, here's where the flow cytometer would come in, as Dominique had introduced us to earlier. Also some cell type separator technology, particularly if this is coming from a patient where you have different blood cell types that might need to be separated. And then Maggie, if we click again, we can see on the gene-based therapy side, as we talked about, insect culture could be helpful here. So the materials you would need for that. And sorry, I must have animated these as separate bullet points. Stirred plate tank bioreactors, if you're doing mammalian cell culture for the AAV production. And if we can click a couple more times here, thank you. And then gold star means optional. If you want to go that extra mile, electroporators and fermenters for microbial fermentation. For gene-based therapy, here we're talking about producing the DNA plasmids and the material that will then be encapsulated into the virus capsids. So we can move on to downstream processing. Again, here's going to be equipment that's going to be very familiar to anyone who's teaching lab-based skills, conductivity, pH meters, centrifuges. So if we're looking specifically at downstream for both cell and gene-based therapy, again, we're looking at typical downstream equipment, centrifuges for clarification. If you have a disc stack centrifuge, that's preferable. Depth filtration systems for clarification, if you want to do it that way instead. TFFs, and then liquid chromatography should be the last one here. And then if you have bulk filling equipment, again, that's extra and fantastic. So not very different from traditional biomanufacturing. If we're talking about cell-based therapy, there really isn't a downstream process because the cells are the product. So you're kind of done after the upstream. On the gene-based therapy side, if we click a couple times here, like Izzo just said, chromatography system is going to be important for purifying those viruses. Tangential flow filtration will be helpful for concentration formulation, and gold star if you have a homogenizer for that cell lysis step. So fill finish is all kind of gold star for gene therapy. Many of us don't have a lot of fill finish equipment, but if you do, it's again, just a plus. Fill machines, a freeze dryer, lyophilizer. Here's just an image of an isolator that I found super interesting because it's all single use. So that whole bag system just gets thrown away and then you set it all up again. So there are, isolators that are permanent, or maybe you can create some sort of temporary isolator using plastic bags just to show students what that might be like. So we can go on to process support. Again, this is going to be buffer and media prep. So things that you would normally use for buffer prep, and filter integrity tester would be helpful, although there are manual ways that you can test filters as well. And then on, if we click a couple of times, Maggie, we have other standard equipment. Again, you likely have autoclaves and glass washers already. Clean in place, gold star. Most of this is single use, so you don't really see a lot of clean in place equipment, maybe in the microbial fermentation side when you're producing the plasmid. So it really depends on the scale. We can move on to analytical. Most of you will probably have gel electrophoresis systems that can be used for STS-Page, for example, a plate reader. And as Isso was just orienting us to qPCR, a lot of companies are moving to digital droplet PCR, as Jim had introduced us to earlier. The main difference between them is that digital droplet is an absolute quantification process. qPCR is comparative. You have to have some sort of standard that you're comparing to for your quantification. But qPCR are way less expensive, so most places are still using that. And then as Maggie and Jim discussed, endotoxin, bioburden, ELISAs, whatever equipment or tools you need around that. Protein quantification, we looked at the capillary electrophoresis. So, just a variety of small instruments and tools that would be used to quantify these various things and for your microbial monitoring. Then I think we have one more slide. Yep, so on the cell-based therapy side, again, flow cytometry is going to be important. And if you want to even go further, you can do FACS analysis. And then on the gene-based therapy side, we have HPLC, which Isso just talked about, to look at empty versus full capsids. But again, that was another one of these instruments that industry said gold star, not required, but would go the extra step for that. Thanks, Maggie.
Thank you, Barbara. That was great. I think we finished just right on time, but we're happy to stick around and answer any questions that have come up about the hands-on curriculum or the equipment. Let's see. I haven't been monitoring the chat as I advance the slides, but did we get any questions in the chat? And does anybody would like to just ask a question? That's fine too.
I don't see any in the chat, but if anyone would like to unmute.
I just want to say, nice job.
Let me, I'd like to close this by saying that this is something I think will really interest students, that this is a series of therapies that are going to alleviate human suffering. If you add stem cells to this, it really is, the blind will see, the deaf will hear, the disabled will walk, children will be spared a horrible outcome from their genetic disorders. And our upcoming students, a lot has been said about them, but one thing that I have found to be true is they don't merely want to pick up a paycheck, they want to make a difference in the world. And this is a way that they could. This project's been very interesting, and I wanted to thank the team. It's been a pleasure working with all of you.
I agree. It's been a great team. It's been just over a year, but we've got a lot accomplished. We really got a lot done. So thanks everybody, and thanks everyone for listening today.
And thanks Innovate Bio for
Yes.
hosting.
If we have a moment, we did have one question about filter integrity testing, and wondering if we could provide some clarity on that process. And Maggie, I imagine we must have the filter integrity testing SOPs on the biomanufacturing.org website from the past, correct?
Yes, we may do. Yes. And yes, I would agree. And also, you know, in the textbook, there's explanations in the textbook, Introduction to Biomanufacturing textbook as well.
Thanks.
And we should make a plug that part of this project is we're writing 2 new chapters for the textbook, Introduction to Biomanufacturing, one on gene therapy and one on cell therapy.
I think there was a question from Kisao earlier. I'm not sure if he's still here. He wanted to talk about SF9 versus HEC293 cells, but perhaps he had to leave.
No, I'm still around. Yes.
Oh, hi.
Yeah. Hi. Yes.
Well,
So the question was about if you have to choose those 2 type of cell line, which one will you go? Will you prefer?
Mm-hmm. Well, there's advantages and disadvantages. I'm just going to say for Isso has really come a long way with the HEC293 cells in this short period of time, because all the reagents were available for him to grow the cells, the 3 plasmids for the triple transfection. So he really has developed that curriculum along the way so far for GFP. For the SF9 cells, not everything is that readily available, although those cells are biological safety level 1. And that was really the reason that Jim and myself decided to go with SF9 cells, just to keep that BSL1, and also to give, when we developed the curriculum, to give programs the choice to go with HEC cells or SF9s. I probably will adopt both. Actually, I'll work with Isso to adopt his HEC293 curriculum, and we'll also work up the SF9.
Okay. The reason why I'm asking this question is if you look at real manufacturing companies and so on, most of them, they use the HK cells.
They do.
Yeah,
Yes.
they use HK cells.
Yes.
So for training, maybe
one of our
for
subject
students,
matter experts.
Yes.
Was say that every company that's sending students to uses insect cells.
Yes.
So locally here, it's dominated by insect cells.
Right. So okay.
So we did ask that question specifically. Is it relevant? I remember Jim asking this question. Is it relevant for us to develop curriculum around insect cells? And that the answer was yes from our industry partners.
Mm-hmm.
And that's why we decided to do both
Okay.
for the project.
Mm-hmm. Well, thanks, Maggie. Thanks, everyone.
Thanks, Jim.
Thanks.
Thanks everybody for joining us today. And thanks again, Linnea.